is far, far greater than the number that we know of from evolution. And I think as time goes on from now, more and more of the proteins that we use and know about will be proteins that are completely made up on the computer from first principles rather than variants or things that came through the evolutionary process, which after all is a sort of a random historical accident in a way. This is very much like technology outside of biology, where if we want to solve a problem like build a building or build a bridge, we don't look around in nature for trees that have the right shape. Or if we want to build an airplane, we don't try and modify birds. Instead, we understand the principles, and then the principles of aerodynamics, for example, and then we build airplanes. I think we're now at that transition with proteins. In nature, proteins are primarily limited to the 20 natural amino acids. But of course, there are many, many other amino acids that are possible. It's just that you can't make, it's hard to make proteins with those amino acids in biology, because biology just uses 20 amino acids. So we were interested in going beyond nature's 20 amino acids, and we focused on smaller proteins, specifically peptide macrocycles, because we could synthesize those chemically, and then in that way we could use more than 20 amino acids. So in this case, it's a very similar concept. We generate peptide backbones on the computer, and then we find sequences whose lowest energy state are those backbone structures, And then we make the peptides in the lab, and we solve their structures, in this case by NMR spectroscopy. And what you see in this slide is, I think it's going to be too small for most of you to see. They're the amino acid sequences here. The important point is that when we determine the NMR structures of these peptides, we find that they are very similar to the design models. So the computational model is in green, and the peptides are in the NMR ensemble is in gray. So we can design these very small peptides. These are 7, 8, 9, and 10 amino acids very accurately. And we believe that we have completely enumerated the set of structures you can make from L and D amino acids. So in this case, these structures are made from about half L and half D amino acids. They're not held together by hydrophobic packing because they're too small. Instead, it's torsional restraints, like the Ramachandran plot restraints that come about from strategic placement of proline residues and L and D amino acids, which have mirror image Ramachandran plots, of course. So we can design a wide variety of these macrocyclic structures. Some of you are probably familiar with the drug cyclosporin, which is an 11 residue cyclic peptide that looks like one of these. That's a natural product. Now we understand the principles for designing the complete class of structures you can make in this way. So we're designing these macrocyclic peptides to bind to targets of interest. To be really useful, we would like to design them so they can cross membranes, because then you could take them as pills or they could get into cells. There's been a grand challenge of designing molecules that can get across membranes. The big advantage we have with these molecules is that we design them to have specific structures, and so we can test out hypotheses for getting across the membrane. So we designed, Gaurav Bhardwaj designed a series of macrocyclic peptides where all of the NH groups were making hydrogen bonds within the peptide or were N-methylated. And he solved the structures of a bunch of these, and these are now crystal structures, and they come out virtually identical to the design model. It's really kind of amazing how close these fit. He then used a standard Transwell assay where you have an epithelial cell layer and you add your peptide on one side and you measure the amount of peptide that comes out on the other side. And the higher the bar here, the more the more is getting across. So this red dashed line is the level at which compounds are considered membrane permeable. And many of these peptides, this is just a subset that Gaurav has designed, are getting across membranes quite well. So we can design this new class of molecules and they get across membranes. So now we need to put it all together and design in binding functions. And then that could be a new class of orally available drugs. So now I want to turn to protein binding. And the example I'm going to give you is of a protein which binds subunits of the interleukin-2 receptor. So interleukin-2 is a central immune modulator. It has 3 receptor subunits, this green, a beta subunit, a gamma subunit, and an alpha subunit. And this is human IL-2 here, which binds all 3 subunits. Now, there's been a lot of interest in using IL-2 as an anti-cancer drug, but the problem is that there's been a lot of toxicity, and it's thought that interactions with this CD25 protein, the alpha subunit, are the problem. So we designed from scratch a protein which is not related to IL-2, but has binding surfaces for the beta and gamma subunits. It does not have any binding surface for the alpha subunit. And this is the crystal structure of this protein called neoleukin-2, bound to the beta and gamma subunits. So when we do binding experiments, we find that this protein, first of all, human IL-2 and derivatives of human IL-2 all bind the alpha subunit, that's shown here, but this designed protein doesn't bind the alpha subunit at all. We didn't put in a binding interface for the alpha subunit. Whereas for the beta and gamma subunit, the alpha subunit, this design protein binds very tightly, actually more tightly than human IL-2. And in mouse models of cancer, this design protein is much more effective in curing disease than is IL-2. And so we've actually just started a company to try and bring this into the clinic. Since this is a very stable protein, we can go further. There's a lot of interest in targeting immune activation to specific subsets of cells so you don't cause systemic immune activation. So in this case, what we've done is to split this design protein into 2 parts, and then we can target the 2 parts separately to cells based on surface markers. For example, in this case, we're targeting 1/2 to cells via one marker, and then the other half to cells via another marker. And so only cells that have both of these markers get the 2 halves, and so get interleukin-2 activity. And that's illustrated here. So there's no interleukin-2 receptor activity, binding activity on any cells except for the ones that express both markers. So this is a powerful way to target the activation of the immune system to a very specific location in the body. So I've talked about proteins that bind to proteins. Now I'm going to talk about proteins that bind to small molecules. The example I'm going to give you is of a de novo designed fluorescent protein. So this is green fluorescent protein, which many of you are probably familiar with. It's 238 amino acids, and it's a fairly large beta-barrel protein with the chromophore in the middle. So we thought we tried to design a much smaller protein. This is now only 110 amino acids. You can see it's it's still a beta-barrel, but it's a much smaller beta-barrel. And this protein is, again, designed completely from scratch. It has no sequence homology to GFP or any other protein. The interactions that GFP makes with the chromophore are very different from the interactions that this designed protein makes with the chromophore. So we made this protein in E. coli, actually a couple different variants of it, and it's a little hard to see, but this is now a superposition of the design model on the crystal structure of this. This is the small molecule bound to the protein. And it's not fluorescent in solution, this molecule is not, until you add the protein, and then it becomes the fluorescent. You get this huge fluorescence enhancement. And I wonder if it's a little bit. Is there a way of turning off the light a little bit on the screen? It's a little bit hard to see. When you put this protein into E. coli or yeast or mammalian cells, the green fluorescence is very clear. So we can design new proteins that bind different types of fluorophores. Is that better? I think that's a little better. Maybe it's just weak. Okay.
If I might ask a question,
Yeah.
what guided the design?
So what guided the design is there were two parts. One is that we wanted to design a structure which could completely surround the chromophore and hold it in a planar conformation, because that's what's key to activating the fluorescence. So we had to design a protein which packed on the small molecules sufficiently strongly to keep it in a planar form, and we had to bind it reasonably tightly. And we had to figure out how. We reasoned that a beta barrel structure would be a good way to do this. So actually the hardest part was learning how to design beta barrel structures from scratch. We've now been able to adapt these beta barrels. Once we figured out how to design one beta barrel, we could design many beta barrels, and we're now designing them to bind a variety of different ligands. And yeah.
Like you mentioned that there are so many conformations for a given number of.
Yeah.
How does it folds into the structure you really want to?
Yeah.
This is done in computer.
So we do it. So the way that we're doing this is on the computer. We're coming up with a sequence that that has a very, very deep free energy minimum at the design target structure. So even though our computer model isn't perfect, it's close enough to reality that when we actually make the sequence, it folds up to that very same state.
And how do you know that's really the free energy minimum?
Well, the best proof that it actually is the free energy minimum is that when we solve the structure, that it folds to that structure. You know, it's kind of a proof by construction in a way.
So a related question, like if I take a given protein.
Maybe I should wait. I'm just worried that people can't hear your questions. So I think afterwards we can, with a microphone, people will be able to hear you. So now I'm going to talk about designing transmembrane proteins. So far I've talked about soluble proteins, but now, as you know, many proteins live in the membrane. So if we're going to design membrane proteins, we need to change the physical model, because the way that we're computing energies is we have a model, we have an energy function which describes van der Waals interactions, hydrogen bonding interactions, electrostatic interactions, and repulsive interactions within the protein chain, and then interactions with solvent, with water on the outside, using an implicit sort of continuum solvation model. Now, for modeling membrane proteins, we need to change the model to an implicit model, solvent model now for lipids in the transmembrane region. So we did this, and now we can use the same principles to design membrane proteins from scratch. So here is a design protein with 8 transmembrane helices, and so we express this protein in E. coli, and it goes to the membrane of the E. coli cell, and we can then purify it out of E. coli and detergent and solve the crystal structure, and it's very, very close to the design model again. Here's this depiction of the structure. It looks kind of like a frog with legs and a head. This is the part that's in the membrane. And this just shows the superposition of the crystal structure on the design model. And you can see, again, it's very, very close. So we can design proteins in the membrane with quite a lot of accuracy. And so now we're designing proteins with membrane proteins with functions. And the most obvious function you might think for a membrane protein is to have a pore in the middle. And this shows a series of designed membrane proteins with increasing size pores. Now, if they don't have any pore at all, then they don't let any ions through. But if they have pores that are several angstroms across, then we can express them in mammalian cells and do patch clamp experiments. And this shows data on this pore where it's not letting sodium through, but it's letting potassium through. And this is interesting. We didn't really design this to have any intrinsic ion selectivity at this stage, but we're observing the physical properties of the pore. Now, when we go to them in mammalian cells, they kill the cells, but miraculously we can still make them in E. coli, and we can also produce them by in vitro translation, and they incorporate into liposomes, and these ones with larger pores allow small fluorescent dyes to pass through, and that's what we're measuring here, the amount of fluorophore getting from the inside out to the outside of a liposome, whereas the ones with the smaller pores do not let fluorophores out. So we can not only design membrane proteins, but we can start to design membrane proteins with channel activity. Okay, so for the next part of my talk, I'm going to discuss proteins that interact not with different small molecules, different proteins, but interact with other copies of themselves to produce one-dimensional or two-dimensional or three-dimensional materials. And the first example I'm going to we take the proteins to make interactions with each other to direct the formation, to make the fiber arrangement the lowest free energy state. Then we make these proteins in E. coli, and again, it's hard to see, I'm sorry, but we can solve their structures of these proteins form fibers in E. coli. So someone should ask me after my talk what fraction of designs really work, and I'll tell you the truth. Right now, I'm being totally dishonest during my talk because I'm just showing the things that work. But we were able to solve the structures of 6 of these fibers by electron microscopy, and you can't see the actual images here, but you can see this is a cross section and an edge-on view of the design model, and this is the same thing for the cryo-EM structure. So you can see that they're pretty close, and superpositions of 3 adjacent monomers on the cryo-EM structures are shown here, and again, they're pretty close. So we can design these proteins that self-assemble into one-dimensional fibers with quite reasonable accuracy. And when we express them in E. coli, the E. coli do interesting things. So E. coli are normally pretty short. This is a 5-micron scale bar, and you can see that when we produce these fibers in E. coli, they form these very, very long cells. So we sort of think of these as synthetic cytoskeleton. And things like microtubules have organizing centers. We can create something like an organizing center by designing what we call anchors, which position monomers of the fiber in an arrangement which shown here, to which additional monomers can bind. So if we make such anchors and we coat magnetic beads with them, and then we add the monomers, the fibers grow off these beads. But if we just take magnetic beads that don't have the anchors on them, they don't grow off. And if we put these anchors on to cover slip and then add fluorescent monomers, we see very rapid growth of the fibers coming off the monomers. So we're now designing proteins that bind to these fibers, and ultimately we're interested in making proteins that can walk on these fibers, molecular motors. So that was one dimensions. Now I'm going to tell you about two-dimensional materials. And in this case, the material is made out of 2 different components, a magenta protein and a green protein, which are designed to come together to form a hexagonal lattice, as shown here. So when we make these 2 proteins in bacteria, and then we simply look at what's formed in the bacteria, we see this extensive hexagonal array. If we purify the proteins out, so we have the 2 proteins in 2 different test tubes, they don't form any type of structure in solution. But if we mix the 2 proteins together, they form this very extensive hexagonal lattice shown here. And if we take an average of this average of of this lattice, we get this density shown here. The computer model, which I showed you before with the two different proteins, is shown here. You can see it fits really, really well on this density. And these lattices can be very, very large. If we just take either one protein or the other, we don't get any assembly. They don't have interfaces that can interact with each other. But then when you mix them together, you get this huge lattice. So if we form these lattices and we put binding proteins for cellular cell surface proteins, then basically what happens is, you can see here reconstitution of just a 3D view of one of these patches binding to a mammalian cell. And basically, let's see, what happens is that the patch is labeled with GFP, and in the cell has a cell surface protein which binds GFP, and all of those cell surface proteins cluster underneath the patch. So this is a very general way of getting a cell to be polarized from the outside. You add a patch, and you can put binding domain for anything you want coming down from the patch, and all of the receptors for that binding domain will get localized underneath it. So we're very interested in biological applications of this for cell polarity and differentiation. So that's a two-dimensional material. And now I'm going to talk about three-dimensional materials. And I'm going to focus on these icosahedral structures, which are now made from two protein subunits, a gray one and, in this case, a gray one and an orange one. And these have designed interfaces which guide the formation of these icosahedral structures. You can see so in this case, one of these proteins has a his tag on it, and the other one isn't. So when we make the two proteins in E. coli, we purify them on a nickel affinity column, and what comes out are these very homogeneous populations of particles. So we can average these particles, and we get these views down the different symmetry axes. Each one of these is a different design, and you can see that the averages, projection averages we get are very similar to what you get from the computational models. And we were able to solve crystal structures of 3 of these, which are shown here. Here are the design models, and here are crystal structures down the different symmetry axes of an icosahedron, a 3-fold axis, a 2-fold axis, and a 5-fold axis. And you can see that the designs are very close, the crystal structures are very close to the computational design models. So this is pretty neat, because I showed you before that for those very small cyclic peptides of 7 or 8 amino acids, we can design sequences that folded up to very unique structures very accurately. And now here you see that the same methods allow the design of protein, of assemblies with thousands of amino acids that are many megadaltons in size. Now I'll talk about various applications of these to vaccines and other areas in one of my later talks. Today I'm just going to talk a little bit about the challenge of targeted delivery, which is sort of a grand challenge. Can we target, say, nucleic acid to a specific location in the body? So as I said, we've designed proteins to bind to many cell surface proteins. In this case, this is a small protein that binds to alpha V beta 6 integrin. If we take this protein, we put a fluorescent label on it, and we put it into a mouse with 2 tumors that have been implanted, one which expresses alpha B integrin and one which doesn't. This protein goes to the tumor that has the alpha V beta 6 integrin, but not to the other tumor. If we display this protein on the inside, RNA are made from 2 different protein components, a blue component and a green component, and when the RNA goes into the cell, or is made in the cell, it directs the expression of the blue protein and the green protein, which now come around it and assemble like in a virus particle to create this protein RNA assembly. Now what we can do is make a library. We can introduce variation in the genome, and then each E. coli cell will make a different type of particle And then we can then collect the particles from many different types from many different E. coli cells and treat them in various ways, like with ribonuclease or with heat, or put them in blood or put them into a mouse, and then recover those particles which survive by reverse transcription followed by PCR, and then retransform into E. coli. And so we can evolve these synthetic particles to have many new types of properties. And so, for example, we were able to very easily evolve them to protect their own genome and ultimately from RNase, and ultimately to circulate for a long time in the body. And so, so moving forward, we're using a combination of design plus selection to evolve particles that can get into cells and release their messenger RNA. One of the grand challenges that I haven't described yet is how do we get these particles out of the endosome? And I'll talk about that at the end of my talk. Okay, so now I want to talk about designing proteins that can do logic operations and can move. And to do this, we took advantage. We're inspired by DNA. So DNA is held together by arrays of buried, centrally located hydrogen bond in groups, the Watson-Crick base pairs. And because of that, it's very easy to design new DNA interactions between DNA molecules. You just write down a sequence, and then it's Watson-Crick base pair complement. But you can't do this with proteins because proteins are usually held together by hydrophobic interactions, not hydrogen bonding interactions. But Scott Boykin, a postdoc in the lab, developed a method for designing proteins which are held together by buried hydrogen bond networks. And so Zibo Chen used this method to design proteins, heterodimers, here shown, this is one chain, a blue chain, and a green chain and a blue chain. And these horizontal squares show the designed hydrogen bond networks. So this one, the yellow are the hydrogen bonds. So it goes from a tyrosine to a glutamine to a serine to a tryptophan to another serine. This is the crystal structure of this design. You can see that designed hydrogen bond network is there. This is a side view. And so Zibo designed a whole series of these heterodimers that are held together by different types of hydrogen bond networks. Here's another one shown here. So to see whether these hydrogen bond networks enabled orthogonal interactions, what Zibo did was he made 16 different designed heterodimers with different hydrogen bond networks, and then he expressed them separately, purified them, and then mixed them together and denatured them so that they were all monomers, and then allowed them to renature. So here, any one of them can go back with any of the others. He then, in collaboration with Vicky Wysocki's lab, used native mass spectrometry to see which pairs actually formed. And in this plot, this is all 32 chains by the same 32 chains, and the colors indicate which species are actually forming in solution. And in red squares are the designed ones with the complementary hydrogen bond networks. And you can see almost everything that was observed in solution was the designed heterodimer. So now we can use this method to make an almost unlimited number of heterodimeric proteins. And Zibo is now using this to create logic gates. So in this example, he's fused one half of a green heterodimer to a DNA binding domain, and one half of a blue heterodimer to an activation domain. So the green doesn't interact with the blue. So this is in a yeast cell, and for it to grow, the activation domain has to be hooked to the DNA binding domain. So then he expresses in these cells the blue complement here hooked to a red heterodimer, or the red complement hooked to the green complement of this one. And in none of these cases do the yeast grow. But if he expresses both of these, then the blue connects onto the blue, these two red connect, and this green connects to here, and you get growth of the yeast. So this is AND logic. I'm going to skip over it, but he's used this to design many different types of logic operations using this basic principle. So I'm now going to talk about the design of allosteric changes in proteins. And the basic principle is that we can design proteins that have suboptimal hydrogen bond networks that can then be displaced by a higher affinity protein interaction. So in this case, we've caged a binding motif for, in this case, it's called BIM. It binds a protein called BCL2. So the data is shown here. We have the target protein on a chip. And if you just add this protein, even though it has a binding domain for this target, you get no binding at all. If you add what we call a key, if we add a key that's short, we still get no binding. But as the key gets longer, then it can start competing out this interaction. This flips up, and we get binding to the target. And that's shown here. So with the long key, it comes in, displaces the binding motif, and we get binding. So we can, this is basically allosteric activation of protein function. We can get binding activity only when we add a second component. So this works inside cells. So in this case, what we've done is to cage a sequence called the degron, which causes proteins to be degraded. So we have different cage key pairs. It's like having different locks with different keys. So in this case, we have caged the degron in what we call cage A. So if we express, so the protein, what yellow fluorescent protein accumulates because the degron is caged, so the protein doesn't get degraded. If we express this key A in the cell, though, it opens the lock, the degron gets exposed, and the yellow fluorescent protein gets degraded. But the red fluorescent protein, which has a degron which is caged by a different cage, cage C, stays totally flat. On the other hand, if we express key C in the cell, key C does not open cage A, so the yellow protein is fine, but instead the red protein gets degraded. So we can now regulate protein function through this allosteric activation in cells. And I mentioned before the problem of getting proteins out of the endosome. And so in this case, Scott Boykin designed hydrogen bond networks here between three helical hairpins. There's a blue one, a green one, and a magenta one. And these are, each one of these networks, and this is one example of a network, there are two others in this protein, has 3 histine residues. So these histine residues both make one hydrogen bond, and they accept a hydrogen bond from one residue, and they donate to another. So this is a very stable trimer above pH 7, but below pH 7, the histines get protonated, and this whole thing breaks apart. And you can see that by mass spectrometry here. So the trimer starts falling apart very cooperatively just below pH 6. Now this is interesting because this is the pH of the endosome, and so we wanted to know what would happen with these proteins and liposome disruption. So this is an assay in which you have liposomes which are filled with a fluorescent dye, and when we add the proteins, at pH 7, nothing happens. The dye molecules quench each other. But at pH 6 and below, this whole structure comes apart, and these peptides insert into liposomes, and the dye rushes out. So what we're doing now is putting this protein on the surface of those cages, encapsulating their own RNA, and we hope in this way that we can break out of the endosome. And I have one final example. In nature, there are many materials which are hybrid materials made out of protein with inorganic compounds like bone and teeth and seashells. And we're very interested in designing such composite materials made out of proteins and inorganic materials. So in this case, this is one of those repeat proteins I showed you at the very beginning, which we've designed to have a periodicity which exactly matches that of the mica inorganic lattice. So mica has a layer of potassium ions on its surface, which are shown in purple here. And this design proteins has an array of carboxylates, which perfectly matches geometrically the mica lattice. So we've designed this protein to be very long. It has 19 repeats, so it's 900 residues long. And we can actually see it by atomic force microscopy. We can see how it binds to the surface. And you can see it binds in very oriented ways. So we can design interfaces between these. These are the repeat proteins that have the carboxylate, the glutamate residues, and aspartate residues that are interacting with the lattice. And we can design interfaces between these to direct the formation of different types of assemblies. So here we've designed trimeric interfaces between these that direct the formation of these hexagonal grids. And the nice thing about repeat proteins is we can just change the grid spacing by changing the number of repeats. And that's shown here. This is going from 3 repeats to 7 repeats. And you see how the grid spacing gets larger and larger. It's going to be hard for you to see, but when we put this protein down on a mica surface, we indeed form the hexagonal lattice. You can see this is the averages of the experimental AFM images, and you can see they're very close to the design models. And we can really tune in a very precise manner the lattice spacing here. So if you need picoliter size wells on mica, we know how to make them. And so in conclusion, I think I've shown you that we understand enough about protein folding and assembly to now design a wide range of brand new proteins and assemblies. And there's a lot of exciting opportunities for designing new functional proteins to address modern day problems. And I think there are people working, you have their names here, and
So, yeah, an expectedly spectacular talk. I'm sure there are lots of questions. So I'll just
again, I think the best bet for that are these macrocyclic peptides. So cyclosporin is an orally available drug. You eat it, and it goes into your bloodstream. And I showed that these peptides can get across basically a model of the gut epithelium. But we haven't yet designed things that would be useful as drugs. But I think that's sort of a very exciting frontier.
So what happens if you bring in post-translational modifications into the picture?
Yeah, so one of the post-translational modifications we have looked at, which I'll talk about in one of my later lectures, is phosphorylation. And we can make similar types of allosteric switches by using kinase phosphorylation. And that's most of what we've looked at for post-translational modification.
Yeah. So it is a fantastic magic show, but unlike the magic show, these are real. It's really done. I mean, my question is, when you say we come up with sequences, which are not, say, similar to the natural protein sequences, what is it in your computational algorithm that enables you to come up with those sequences to fold into a particular structure?
And I'll talk about that more in one of my later lectures. I didn't want to go too much in the technical details here. But basically what we do is once we have a protein backbone, we can enumerate all the rotameric conformations of all the amino acids at each position. And then it's a combinatorics problem. It's like a jigsaw puzzle. We have to find that combination of amino acid conformations and identities where we get really, really low energy. And we can solve that, it turns out, very rapidly by Monte Carlo simulation. And so I'll show a movie of that in one of my later lectures. But it actually turns out to be a pretty straightforward calculation.
It's a sort of, I'm here.
Yeah.
It's fascinating as it is, but one naive concern would be that the synthetic proteins, when you put them into animals, won't they?
A couple things I can say about that. First of all, for some of these design proteins, for example, this IL-2 mimic, we've looked very hard for immune response. very stable, and very soluble. So they're simply not displayed very efficiently on MHC. Now that doesn't mean that when you get to a human that they won't produce immune responses. It's just that we haven't seen them yet. The other point is that if you do get an immune response against these, then it won't cross-react with your own host proteins. So there's a lot of work now or interest in IL-2 derivatives where they make a couple of mutations. In that case, if you get an immune response, it could really be catastrophic because it could interact with your host protein. The one place where we responses, as you might expect, are with hydral cages, and we're viral glycoprotein. And I'll talk about that in one of my later.
But I'm
So I think
and especially students.
Looking from the back, I couldn't see, but
7 to 14 amino acids have no regular secondary structure. So that was one of the things we were excited about, that we could use the same principles to design proteins without any regular secondary structure.
So is there any distinguishable between successful design pool and those that were not successful?
Yeah. So in fact, that's one of the things that we're very excited about with this large numbers of designs using these oligonucleotide arrays, because we can make now tens of thousands, and we can start to learn what the differences are between the designs that are successful and the designs that aren't. And then we can incorporate that knowledge into the design method and iterate. And we found that for both designing stable folding proteins as well as proteins which bind to other proteins, we can systematically improve our performance in that way. You can start doing machine learning on the data because we can generate enough data.
So, for the natural protein stability and their, motion dynamic is very important.
Yeah.
If your criteria is that you design a very stable protein, I'm wondering if all your protein are very, very stable, the one you design.
Yeah. So,
And how
well,
do you
design. I didn't talk about designing sites inside that, say, of catalytic residues which are charged. Then the proteins become.
How often and still retain the same affinity for the.
Let's see. Well, in general, when we're designing binders for before the thing that really made all this possible, and everything I showed you is really the last 4 years or so, was we required that we said we would never make that's often the hard.
Got a protein which replicated the function, but the structure was quite different.
Oh yeah, that's a good question. That doesn't usually. I'm trying to think if that's happened. I think since we've been designing proteins de novo, when we get crystal structures, they are almost always very, very close to the design model. In some cases, we see. It's interesting when they're not, because we learn what mistakes we made. So, for example, we made recently a beta alpha protein where one of the crystal structures, one of the beta strands was sort of slid in register. And studying that, you could see what the principle we had missed was. So sometimes, rarely, the structures come out differently, and those always teach us a lot.
Okay, okay, one last question.
Speaking of beta barrels and symmetry, you had previously designed a 4-fold symmetric TIM barrel, which obviously folded and you got a structure. In that particular case, symmetry worked for you, whereas for the GFP, it didn't. Could you comment on the differences? Yeah,
that's a very good point. It's really a difference in the detailed way in which the strands interact with each other. In the TIM barrel, the strands are all parallel to each other, and it turns out cylindrical symmetry works. In the beta barrel, they go up, down, they're anti-parallel, and it just turns out you can't get perfect hydrogen bonding with a perfectly symmetric structure. And really, it comes down to the detailed stereochemistry of the protein chain. Let me just say, so I'm going to be giving 2 other lectures, I believe. One is going to be more on therapeutics and vaccines, and the other one is going to be on protein structure prediction and and protein-protein interaction prediction, and a little bit more on how we actually do the calculations.
Okay, and let me just wind up with one last question. Now, you know, the hydrogen bonding that you showed for those heterodimeric proteins, you're competing with 55 molar water.
Yeah.
And so getting that exactly right must be exceptionally challenging. Are there other features of the design which are also positioning the group so that they form, or you really have to get the hydrogen bonding spot on independently?
Right. So the hydrogen bonds, I think, are giving you the specificity. They're not giving the affinity, because after all, when those are coming together, they're losing all the interactions with water. So the affinity, the other layers in those helical bundles they're closely packed hydrophobic interactions. So one way to think about it is they're coming together because of the hydrophobic interactions, but if the hydrogen bonds are not complementary, then you'll have this huge loss. It'll be extremely unfavorable because you'll be burying all these polar groups. So basically, what those hydrogen bond networks are doing is they're kind of like gatekeepers. Things can only come together if they match. So they're not really driving the binding reaction. They're providing the specificity.
Yeah, but if they didn't match, then they would take. So I mean, how accurate does one have to be in positioning those atoms correctly so that they?
Yeah, well, for the most part, I showed you the structures we have. For the most part, the hydrogen bond networks come out pretty close. Sometimes we get water molecules inserted in, and so so that's usually the error, that where we thought there was going to be a side chain hydrogen bond, a water would intercalate in.
Okay, so we'll wind up here now. The undergraduates, which I presume there's some, after we have tea, we'll have a session where David can meet them. So once again, it was really a great opportunity for all of us. And before we wind up, I just need to give David a small memento, which is a memoir and biography of C.V. Raman that he doesn't need to put in his suitcase. We'll mail it to him later. Please join us for high tea outside. And after that, we'll come back in.
Yeah, I went a little bit long. I didn't know what time I started.